Okay, now we're ready to get started. Okay, so last time I talked about DNA repair, and I'm going to finish up, about DNA replication. And I'm going to finish up DNA replication today and say a little bit about repair and recombination, most likely. So we'll see how that goes. But wherever I finish today, that is where the exam material will stop. So last time I talked about the initiation of E. coli DNA replication, and I pointed out that it is all DNA replication starts at specific places. Okay. The exception to that is, of course, on the lagging strand, every so often one starts. But the overall process of replication always starts at a specific place. In the E. coli system, it's known as ORI-C. In our chromosomes, we have many replication origins because we have gigantic chromosomes compared to what E. coli has for its. The size of the E. coli genome is about 6 million base pairs, and they have one replication origin. We have about 7 billion base pairs, and so that's more than 1,000 times greater. You could imagine we would have quite a few more origins of replication, and in fact we do. I'm not going to spend a lot of time talking, however, about our origins of replication. Now, as I mentioned last time, the binding of DNA A to these sites on the ORI-C section of the E. coli chromosome causes a winding to occur, and that winding causes some unraveling of the AT-rich regions next to those binding sites for DNA A. The binding of DNA B, which actually comes into something called DNA BC, and then C takes off and leaves behind these proteins here that are helicases, and the helicases actually start stripping apart the strands so the replication can proceed. There are actually, in E. coli, your book is a little in error in that it only shows 3 DNA polymerases. E. coli actually has 5 DNA polymerases. 2 are not called, they're not named as DNA polymerases, and that's probably why they didn't put those there. For our purposes, they don't, the other 2 don't play any big roles. And in fact, for our purposes, the only DNA polymerases in E. coli that we're concerned about are DNA polymerase 1 and DNA polymerase 3. As I noted, DNA polymerase 2 is involved in repairing damage to DNA. It's called an error-prone polymerase, as are the others. And the reason for that is because when DNA is damaged, making guesses about what the appropriate base pair is makes some sense if you're going to try to synthesize a strand. And so the other polymerases tend to be of that nature. Eukaryotes have similar systems to E. coli in the sense that they have lagging strand and leading strand replication. But as we will see, there are some other considerations for replication of eukaryotic linear chromosomes, and I'll say a fair amount more about that. In eukaryotes, there's a lot more orchestration to the overall process. Somebody asked me last time if there is control about when replication occurs and so forth, and the answer is, in E. coli, yes, there is to some extent. But in eukaryotic cells, there is to a much greater extent. Okay? Cells go through what are called a cycle, and that cycle involves synthesis, where DNA is synthesized, and a couple of different phases, G1 and G2, and finally a mitosis phase where the cells actually divide. What we're concerned with is what's happening in the S phase, and I will say a bit about that. One of the considerations in replication in eukaryotic cells is that the DNAs are not circular. That is a very, very, very important point. The chromosomes in eukaryotic cells are linear. Okay? And replication at the far end, where the end of the chromosome is a problem for cells. When people first started studying eukaryotic chromosomes, and they were curious about that linear nature, how it was that the ends got replicated, they noticed something, first of all, very curious. And the curious thing that they noticed was at the ends of linear chromosomes, there were structures called telomeres, T-E-L-O-M-E-R-E-S. And the telomeres, when they analyzed them for sequence, they discovered had thousands and thousands of copies of about an 8 to 10 base pair sequence that was repeated over and over and over and over. Okay? And people looked at that and thought, oh, well, maybe it's just some way of identifying this is the end of the chromosome, and that was the end of the story. Well, it wasn't the end of the story because people started investigating, what the nature of these telomeres were. And what they discovered was something rather interesting. They discovered that as cells got older and older and older, the number of copies of repeats in the telomere shrank. That meant that every time that a cell was replicating, the telomere was getting shorter and shorter and shorter. Okay? And so some people have compared this telomere getting shorter to a fuse on a bomb. That fuse, with every division of the cell, with every replication of the DNA, is getting shorter and shorter and shorter. Eventually, cells get old enough, they're going to run out of those repeated sequences. And when they start shortening other things like genes that are essential that are next to those telomeric sequences, the cell is going to have trouble. The cell is likely going to die. Okay? So we need to think a little bit about those telomeric sequences. First of all, how and why they shorten, and then how it is that they get made, and some other considerations relative to those. There are some very interesting theories out there, and there's some evidence that the longer the telomeric sequence is, that is, the more repeats that the telomeres have, the longer the cell will be alive. Okay? Now, the telomeres that you are born with may play some role in the lifetime or the lifespan that your cells will have. Now, it's not as simple of a picture as I'm painting there, but we could imagine that longer telomeres give more divisions. We sort of knew that. When we took cells out of organisms, and we can scrape cells off of your skin, or we can scrape cells off of your liver, or you pick a tissue, we can do that. We can actually grow those cells on a petri dish for a period of time. Okay? That is, if we feed them the normal nutrients that they would get in the bloodstream and so forth, those cells will continue to grow and will continue to divide. The problem is they won't do it for very long. They have a fixed number of divisions that they will go through before they stop. And one of the things that stops them is the fact that they just run off their telomeres. Well, how come telomeres shrink with each division of the DNA? Okay? Well, this high-quality figure that I drew shows you a little bit about how that shortening process occurs. Here's a linear chromosome that we have as a 5' end and a 3' end and a 5' end and a 3' end. And we see that to start at the very end, and I'm not showing all the other replication that's occurring, but if we start at the very end, we see that we put down an RNA primer that I've shown there in red. There's an RNA primer here in red that goes in this direction. So one's moving towards the right, the other is moving in towards the left. At the end of that replication, we have the following. We have a strand on the top, okay, that has a red primer on the right side, and we have a strand on the bottom that has a red primer on the left side. Okay? Well, for simplicity purposes, I'm only going to follow this top one right here. When this guy starts to replicate, again, what we see is, okay, well, at this end now we get a red, and replication proceeds. However, we have a problem. Replication will only proceed until the replication gets to the red sequence. Okay? So here it's a little confusing, but here, this 5 down here is this 5 right here. All right? Replication continues, but DNA polymerase can't copy RNA. DNA polymerase can't copy RNA. That RNA primer that's there just sits there and never gets copied. And you're sitting here thinking, well, don't cells have a DNA polymerase 1 to remove that? Cells actually do have a way to remove that, but they have no way to replace it. In E. coli, we had a circle, and the circle came all the way around, and when it came all the way around, that coming around part became the primer. There's nothing that can prime out here. So what this means is that every time this chromosome goes through a replication, it's going to lose a little bit of sequence at the 5' end. Now you're probably thinking, well, if that happens, then how come any of us are alive? How come we haven't just shortened our telomeres down to nothing because human cells have been around for a long time? And the answer to that is it turns out that telomeres can be made. And they are made by action of an enzyme known as telomerase. Telomeres can get made. Now I'm going to tell you in a second how that synthesis occurs. All right? But suffice it to say that that enzyme is essential for making functional telomeres, and that's what you saw in this figure right here. This actually shows how this process occurs. Okay? So how does telomerase make linear ends? All right? I'll show you how it works first, and then I'll tell you later why your telomeres are shortening, because you might think, well, it keeps lengthening, we don't need to worry about that. Yes, you do. All right. Here's the end of a eukaryotic linear chromosome. There's the telomere, there's some repeats that are sitting there, and there's a thing sitting off of the end. The telomerase is a DNA polymerase that's odd and interesting. So telomerase is a special category of DNA polymerase. Like any other DNA polymerase, it needs a primer. And the primer in this case is the 3' end of one of the chromosomes. The telomerase carries with it a tiny RNA that serves as the template. That is something that it can copy. I'll repeat that. That's a very important point. The telomerase is carrying its own template to be copied, meaning the other strand to be copied. It's carrying it with it. Well, there's the sequence that gets copied. We see that telomerase copies this RNA that it's carrying around with it. You can see it being copied right there. And once it's copied it, the telomerase scoots it down and copies it again, and again, and again, and again. Every time it scoots it down and copies it, it adds another copy onto the end of that. And if we get this guy long enough, then a primase can say, oh, I'm going to start and go back this direction, and it starts filling in the other strand. So as a consequence of this telomerase, we can lengthen one of the strands quite easily, and then we rely on a primase to start making a short primer, and then DNA polymerase can extend it, and as a consequence, this 5' end keeps moving out further and further and further. Now, why do our telomeres shorten with every round of replication? The answer is actually quite simple. Telomerase is not active in the vast majority of our cells. It's not active. It's active when we are an infant. It's active when we are a fertilized egg. Okay? And when we are a fertilized egg, that telomerase is sitting there going crazy, and it's making copies and making copies and making copies and lengthening those chromosomes on and on and on. After we're born, telomerase, in the vast majority of our cells, stops working. One of the questions people have at this point is, well, if I can activate telomerase, will I live forever? And the answer is, you might. But you might have other problems. Okay? The other problem is that I said that some cells have telomerase active. Not all cells have it inactive. And one of the classes of cells that have telomerase active are known as cancer cells. Cancer cells, if you think about it, have to have something to make the ends longer and longer and longer, because cancer cells are dividing rather uncontrollably. And if they didn't lengthen their telomeres, what would happen? They would die out. So instead of thinking of activating telomerase, what most scientists are thinking of doing is finding ways to inhibit telomerase. Inhibiting telomerase as a cancer treatment is actually in clinical trials right now, and there are some promising results. If you inhibit telomerase in cancer cells, they may divide themselves out of control, and they die out, and in some cases it appears that that's exactly what happens. So telomerase is a very, very cool enzyme and a very interesting enzyme. All right, I'll stop at that point and take some questions. Yes? So yeah, stem cells will have telomerase active. Yeah, so that's pretty important that they have that. You bet. Yeah.
What is the correlation between active telomerase and, cancer isn't just because your cell has one telomere, there's other damage that happen in it, in your DNA. So what's the correlation between having active telomerase and the damage elsewhere in the DNA that's causing cancer?
I'm not sure I understand your question here. It's something to the effect that what's the correlation between the activity of telomerase and damage to DNA? Is that what you said?
Well,
So, okay, so sort of her question is, having telomerase active, is that sufficient to make a cell be cancerous? I think that's part of what you're saying, right?
Well, I suppose in a way. I
Yeah.
know it's not.
Yeah. So, yeah, you're right. It's not sufficient, but we don't know fully what all is necessary to make a cell become cancerous. As you know, there are multiple damaging events that have to happen, so there's no one pathway we can say that this cell is going to become cancerous or not become cancerous. Obviously, if telomerase activity caused cancer, then we wouldn't be born because, of course, we have it active in infant cells and so forth. So I don't have there's not an answer to that question, essentially. Jody?
With telomerase being active in stem cells and embryonic cells, are there any cancers that arise directly from those cell lineages?
Are there cancers that arise directly from embryonic or stem cells? Absolutely. Absolutely. Yeah. Okay, so that's a common question too. So he wants to know how long he's going to live, basically. So, so the question is there an exact correlation between the length of telomeres and human lifetime? And the answer is no. Okay, but you can get an idea of how long, how many generations a cell would be able to remain alive in the absence of telomerase. That you can do. Yes, Jared.
So some tissues have high turnover rate,
Yep. So do cells that have high active turnover rates have telomerase active? Cells that have high turnover rates are going to be more dependent upon stem cells for their function. So in the differentiated cells, no. In the stem cells, yes. Okay. Oh, where? Oh, Connie. Okay, I heard my name. Yeah.
But you said earlier that when cells are growing in petri dishes, they only can divide a certain number of times before they die.
Yeah.
How come humans can, I guess, live longer than cells can?
Okay, so Connie has a complicated question. That is, I said that cells, if I take them off of my body and I put them in a petri dish, they only go for a few generations and then they die. How come we live longer than those few generations that those cells are going? A, those cells in a petri dish are going at an accelerated rate, so they are dividing more rapidly than they would divide if they were in our body. That's one. And number two, again, stem cells are helping to replenish cells, and so that is a big variable that we have a hard time quantitating there. When I put cells and grow them in a petri dish, it's in the absence of stem cells. Okay? Good question, though. Yeah.
Okay, phrase it better.
Okay, so come back on the question.
What is it about cancer, the damages in cancer cells that activates telomerase?
Oh, okay. So the question sort of that she's asking is sort of flipped around now. That is, how is it that cancer cells turn on telomerase? All right. Well, we could think of this as almost a selection process. All right. That is, if they don't turn on telomerase, we're probably never going to see them. All right. So turning on a gene, there's many different ways that we can turn on a gene. We'll talk about that a little bit with transcription later. But we can think about this as being a pretty important event in the genesis of a cancer cell. So how it happens might happen by a variety of mechanisms, but it's almost certainly going to have to happen because if it doesn't, the cancer is going to die itself out. It's probably not a very satisfying answer, but that's what it is. Yes, sir.
So that would be like saying there's more cancer to start with, and the ones that actually survive as the tumor are spontaneous replicates that turn it back on?
Right. So Jody's sort of reiterating what I'm saying here, and that is that there are many different mutations, many different transformations that can happen to cells. And when we see a tumor, there's been a selection that happens by the time we get to that point. Absolutely, that's the case. Okay. Every one of us right now is forming tumor cells in our body as we're sitting here. Every one of us is. And the vast majority of those are never going to see the light of day. The immune system's going to get them. They're going to burn themselves out. They don't have proper things to grow uncontrollably, et cetera. So there is definitely a selection process that's happening in the formation of a tumor. You bet. Yeah. One last question, Kyle.
telomere is very important in cancer growth.
Yep.
Has telomerase ever been examined for cancer treatment?
So that's what I just said. So his question is, has telomerase been examined for a cancer treatment? The answer is yes. There are clinical trials going on where people are using inhibitors of telomerase to stop the growth of telomerase, and so the cancer cells actually burn themselves out. Okay? Okay. So that's telomerase, a fascinating enzyme, fascinating story, and we're still learning an awful lot about telomerase. Well, that completes the last of what I want to say with respect to DNA replication. I'd like to turn our attention briefly to DNA repair and say a little bit about that. Damage to DNA is something that happens as a result of exposure to hazardous chemicals, as a result of exposure to reactive oxygen species that our own cells are generating all the time. So reactive oxygen species themselves have been very actively studied now for several years, and there's an increasing interest in them because the more we understand about how reactive oxygen species are causing damage in cells, the more we understand about processes like mutation that give rise to tumor formation and so forth. What you see on the screen is a very interesting and sort of scary compound. It's called 8-oxoguanine. And 8-oxoguanine is just like guanine, except that it has an extra oxidation that has occurred, and that extra oxidation is here, and results in the formation of a stable base pair between guanine and adenine. As a result of this, if you have DNA that has 8-oxoguanine in it, or if you have 8-oxo-DGTP that DNA polymerase uses, in either case, guanine may be paired across from an adenine. Well, that will obviously be a source of a mutation that happens in cells. And so cells have to have, or ideally have, ways of recognizing and dealing with that. There are, in fact, enzymes that are present in our cells that recognize when damage has occurred to bases and actually removes them. Okay? Other damage that can happen to DNA includes the damage to adenine by deamination, in this case making hypoxanthine. We've seen hypoxanthine before. And hypoxanthine is going to have very different kinds of pairing rules than is adenine. We could imagine if this happens in our DNA that we have problems arising with base pairing. Other damage that can happen to DNAs include chemicals that will covalently bond to structures in the DNA. And some of these are pretty nasty. Okay? Here's one called aflatoxin. Aflatoxin is made by, it's a compound made by a mold. This mold is commonly found in peanuts. And aflatoxin is pretty nasty because what happens is in our body, the body tries to detoxify it, and in the process of trying to detoxify it, creates something that is extraordinarily reactive. Okay? So it's the detoxification, or the attempt to detoxify, that causes a big problem. It creates this little three-membered ring over here that will, when it bumps into DNA, covalently bond to the DNA. So now what we have, instead of bases and phosphates and sugars in the DNA, now we got this big honking ugly thing that's sticking out or sticking inside of the DNA double helix. That causes, as you could imagine, a great big bulge. It causes problems for the DNA polymerase moving along, trying to copy the base pairs. So these are known as alkylating agents, okay? And they are toxic. They can be very mutagenic. They can cause some very severe problems when they get into our DNAs. Another important covalent modification that happens to DNAs are those that are known as thymine dimers. Thymine dimers arise when two adjacent thymines on the same strand are exposed to ultraviolet light. Okay? Well, we get exposure to ultraviolet light all the time. And when that happens, a covalent bond can be formed between these two thymines. Okay? Now, I want to emphasize these are on the same strand, not on opposite strands. They're on the same strand, one thymine next to another, like one above the next one in the ladder of the DNA. Okay? When that happens, this covalent bond between the two thymines basically stops DNA polymerase from being able to function, from being able to go through and complete its replication. Not surprisingly, this can, if it's not repaired, lead to mutation and errors in the DNA as a result. How do we get thymine dimers? Well, one of the best ways to get thymine dimers is to go use a tanning booth. And last, there are reagents that will do what's called cross-linking. That is, they themselves will bond one side to one portion of a DNA and another side to another portion of a DNA. These cross-links can be within a strand. They can be between strands. They can even be between two helices. In any event, if any of those bonds are formed, what happens is, again, a DNA is damaged, and that DNA cannot be copied properly by a DNA polymerase. So for all of these reagents that I'm showing you on the screen, cells need to have some way of dealing with them, because if they don't, they're going to become much more mutagenic. Now, what I'm going to be explaining to you in just a minute is that there are some systems that our cells have to repair these. So it's not hopeless. But I want to also emphasize that the more you are exposed to these sorts of things, the more likely you're going to overload the repair system. So it's not all good news. Jody?
Since bonds are actually formed in this one between the two different strands, are they not considered interchelators?
Are these interchelators or not? This guy probably could intercalate. It's a fairly flat structure. So I would say that, yes, it probably could be an intercalator, but it doesn't have to be an intercalator in order to function. So this could happen, as I said, this could be one chromosome over here, and this could be the other chromosome over here. So it doesn't even have to be inside of a helix. But it could be. Make sense? Okay. Well, so the bad news is that we don't want to overload our system. The good news is that hopefully we have some ways of dealing with them. Well, you've already seen one system for taking care of mistakes that I've talked about, and that was the proofreading system of DNA polymerase. You remember that the 3' to 5' exonuclease activity was useful when the polymerase said, oops, I put the wrong base in, let's get it out of there. And so that's not really damage. We don't have damage that's happening to that DNA. It's simply a mistake the polymerase made. But it's important that the polymerase take care of mistakes that it makes. What about those mistakes that the polymerase doesn't recognize? Okay? So we're saying, here's this proofreader. It comes along. It says, oh, I made a mistake. I'm going to back it back out. But I told you when I introduced the proofreading to you the other day that no proofreading is perfect. Okay? Proofreading by DNA polymerase improves the accuracy of the polymerase by about 100 to 1,000-fold. But even there are mistakes that slip through. Okay? So what happens if a mistake is not caught? Well, it turns out that cells have a backup. They're not reliant totally upon proofreading. Here is a system where, for example, the polymerase didn't recognize that, oops, I put in a T across from a G. Okay? All right? This is what's known as a mismatch, and you can see the polymerase went along its merry way doing its thing. Okay? The system I'm describing to you here is in E. coli, but an almost identical system works in our cells. Okay? So we have a very, very similar system. They have different names, or slightly different names, but a very similar system works inside of our cells. How does this work? Well, E. coli has a protein called MUT-S, M-U-T-S, and its sole job is to scan the DNA and find where mispairs have occurred. How would a protein know where mispairs have occurred? What would be characteristic? Yeah? Number of hydrogen bonds? Well, sort of, but there's a better answer than that. Jesse? A bulge. Okay? Everything else has got perfectly paired up hydrogen bonds and perfectly paired up bases, but now these guys, G and T, the hydrogen bonds don't fit properly, so there's a bulge. So it's very simple. MUT-S goes along and looks for bulges. It finds a bulge and says, oh, look, something isn't right here. Once MUT-S binds, it binds to a second protein known as MUT-L. And MUT-L doesn't do much except for provide a bridge between MUT-S and the active protein, which is called MUT-H. MUT-H makes a little cut right there and then excises back. And after it excises back, DNA polymerase comes along and says, oh, look, we've got a gap got to fill in. Polymerase comes along, fills in the gap. DNA ligase comes in, seals it up, and everybody's fine and dandy. Now, this is a very simple system, and it's a very nice way of proofreading the proofreading. Okay, How about another repair system? This repair system I'm going to talk about is called DNA Excision Repair. The last one was called Mismatch Repair. This repair system is very useful if it turns out that there is something in the DNA that has bound two bases together. For example, I talked about thymine dimers. I said they both link two bases on the same strand, and what you see in this might be an example of thymine dimers. This could be a thymine dimer right up here in blue. Well, when this happens, there is an enzyme called, here's a mouthful of a name for you, UVR-ABC exonuclease. It comes along, it does everything in one. It recognizes, it chews back, it removes the damaged piece, it allows DNA polymerase to come back in, fill in the gap, and DNA ligase fills it in, and everybody's happy again. You'll notice on the last one it said that DNA polymerase 3 filled in the gap, and you'll notice here it says DNA polymerase 1 fills in the gap. I don't care if you know which one that is. I think the important thing is that the gap gets filled in. You should also know, of course, that the DNA ligase is needed to seal that once we've gotten everybody together. Does this mean I can go to my tanning booth and tan all I want? Well, as I say, I think I should be careful with this because I don't want to overload this system. I overload this system, what's going to happen is I am going to cause some giant problems of mutation if all of these don't get fixed. Yes?
So the UVR-ABC and the mute system essentially do the same thing, but the ABC does it in one concerted step?
That's right. So his question is the UVR ABC exonuclease versus the mute SHL system are similar in what they do, and this is doing it, and I'm simplifying it a bit, but this is doing it with one enzyme. That's correct.
So when does the body know what system to do? How does it?
So his question is, how does the body know which one to use? Well, we had two different situations here. The first situation, we simply had a mismatch.
Yeah.
That was a G mispaired with a T. In the second situation, I had a chemical bond that was formed between a couple of bases. So those are fundamentally different things for the cell that it will encounter. Okay? Yes, Lawrence. Mute L attracts the exonuclease. Yeah. Yes, sir. Jared.
Since polymerase is pretty active or pretty efficient at very correctly goes at some odd number of seconds, how accurate are these systems?
Okay. So his question is, how accurate are these systems at catching what they do? Well, it's a little hard to quantitate that if you think about it, because with polymerase, it's fairly easy to measure, let's say, 1 million bases and look to see how many errors do I have. It's a little hard to find 1 million errors like this and quantitate that. So these are pretty darn good. We can tell their accuracy by how much they improve accuracy compared to systems that don't have them. And again, they can improve accuracy of DNA replication by about 10 to 100. So they're part of that overall enhancement that we have of fidelity of replication that I described like for proofreading. Did you have a question behind Jared there also? Oh, yeah, very good question. So his question is, how does the cell know which side to fix? I've got a GT mismatch. Do I take out the G or do I take out the T? Okay, I didn't tell you that, but I will tell you that. It's a very interesting story, okay? It turns out that cells have to know which side to take out, because if they take out the wrong one, then they've just made the problem worse, right? So if you have a 50-50 chance of being right, that's not what you want, because you might as well have the mutation. And it turns out that cells actually can tell which strand to take it out of. It's a little complicated, but I'm going to briefly tell you about it, and I'll tell you about it, and I won't hold you responsible for it. How about that? Okay? In E. coli, we know the system pretty well. E. coli use a methylase. We talked about methylases when we talked about restriction enzymes. And in E. coli, they look for the sequence GATC, and they put a methyl group on, I think it's the A. Okay? For our purposes, it doesn't matter, but they put it on there. All right? Well, GATC is a what's called a complementary sequence. If I go GATC on the top strand, the bottom strand reads GATC the other direction. So I have GATC, I have GATC on each strand, right at the same place, right? When the methylase comes along, it methylates and says, oh, there's an A to put a methyl on, there's an A to put a methyl on, and puts a methyl on both strands. You with me? DNA polymerase comes along and it makes that new strand. What do I have? Before the methyl group methylase gets there, the top strand has an A with a methyl on it, and the bottom strand has an A with no methyl on it. The cell can read that strand and say, oh, there's a GATC with no A on it. That must be newly replicated. This must be the strand to take it off of. And that's how they do it. It's a really cool system. A really cool system that you'll now forget about because I told you didn't have to remember it. Yes, sir?
Is there any pharmaceutical approach to enhancing the levels of these mismatch repair enzymes in humans?
Is there a pharmaceutical approach to enhancing mismatch repair in humans? Not to my knowledge. And I would imagine that would be a very difficult thing to do. How do I make a chemical improve replication fidelity? That's a tough one. More likely, I'm going to define a chemical or develop a chemical that's going to reduce replication fidelity, and that's going to be a problem. So,
Enhance the gene transcription sort of approach?
no. So if you're saying enhancing the transcription of repair genes, you sure would like to do that, but just having more repair genes doesn't necessarily ensure that anything is going to be working better. Okay, that would happen if you had a shortage of these proteins. And so if you're thinking about going to the tanning booth and doing this, then I suppose that might be an approach, but I would imagine you would have other issues that would arise. I don't think that's a practical thing, unfortunately. Okay. All right, we're moving along. One other thing I want to talk about, all right? And the other thing I want to talk about here is getting rid of U's, okay? Getting rid of U's. How do I get U's in DNA? Well, it turns out there's two ways to get U's in DNA. One, I sort of described before. How do you get a U in a DNA? Anybody remember? What's the precursor to get it in? No. DUTP, right? Remember when we were making thymidine, we made DUTP, and I said if we have a little bit of it in this cell, the cell can use it in place of a T, and it'll put a U in the DNA. All right? Well, cells don't want to have that U in the DNA, all right? And so they have a system to remove it. Another way that can happen is if you deaminate cytosine. That is, cytosine has an amine group on it that is chemically slightly unstable, and over time, some of those cytosines will lose their methyl groups and become U's. Well, U pairs like T. U pairs like T. So if I had a C there and it now became a U, when replication occurs, this guy is going to put an A over here instead of putting a G over here when it replicates. So cells want to get those U's out of the DNA, and they have this system that you can see on the screen for removing them. Okay? This involves use of what's called a glycosidase, and glycosidase repair works slightly different to what you've seen before. Glycosidases clip off the offending base from the sugar. So we're actually breaking the bond between the base and the sugar, and that's what's removing that U right there. Okay? That has been removed. There's no base at that point. There's a gap. Well, that gap is a signal to another enzyme to come in, open up a gap like we've seen before, and DNA polymerase comes in and fills it back in. All right? But the first step, and this one differs in the first step, in that the offending base is actually removed from its sugar by, in this case, a uracil N-glycosidase. Okay? Uracil N-glycosidase creates a missing base structure that is a target for an enzyme called AP endonuclease that basically clips the region out and allows DNA polymerase to come in and do its thing. Yes?
So what happens if the DNA gets replicated while you're still in there? Then it used carried from A.
Yeah. So if DNA, that's a good question. So what happens if the replication proceeds before the repair happens? Well, you will have just converted a G into an A, so you will have had a mutation, absolutely. Yep. And so maybe I'm not answering your question. So So let me just take your thing a little further. So let's say we've got this mismatch. Replication comes along, and now it makes a new strand, and it's got A up here paired with a U.
Yeah, my question was if it corrects both strands of that.
It can't correct both strands. It only corrects the U that's in there because the U doesn't belong in the DNA. So now I've got an AU base pair. There's no bulge. It doesn't look like it's a mismatch. But this uracil DNA glycosylase is going to be looking for U's. If it sees U's, what's it going to do? Well, it's a U paired to an A. It clips it out, right? And when the polymerase comes back in, what's it going to do? It's going to put in a T. So we've had a mutation, right? If we don't fix that, we're going to have a mutation. And so that uracil glycosylase is playing a very important function in that process. Okay. Let's see here. How am I doing on time? How about a joke? I haven't done a joke in a while. Okay. This is just a very short joke, but I love this joke. All right. When little fish, okay, are in schools, little tiny fish, they go swimming around in schools, right? When little fish are in schools, their favorite subject? Debate. Now that is rare. I'm rare getting an applause on a very bad pun, but it's not a terrible pun. Their favorite subject when they're in schools is debate.
I think you got a second over here.
What's that?
What about algebra?
You've heard of my joke on that?
No, I say algebra.
That's the second subject.
It's a different subject. I actually have a joke on that, but I can't tell it in class, so I'm not going to do that. Okay, but come by after class and I'll tell you. No. All right. Things are falling apart here. Let's see. What should we do here? Should we? All right. Oh. I know you guys want to sing a song. You want to sing a song? All right, let's sing a song. This is an old 40s song. It's a very hard one to sing, all right? It's called The 3 R's of DNA. I'm probably the only person in this room that'll know this tune. Dream a little dream of me. Base pairs, they all provide you stair steps to form a helix inside you. A pairs with T and G goes with C, making DNA for me. Helicases go unwinding, unzippering at rates almost blinding. Polymerases work night and day, replicating DNA. Proofreading the enzyme's QC path, choose back from the 3's. I can't have a G pair with T, so repair it, please. Chem damage is concerned too, 'cause it can cause mutations inside you. When dimers stem from sunlight UV, fix the DNA for me. Such pathways of excision cause cells to have to make a decision. Should they go straight ahead with repair, or take themselves right out of there? Then lastly, there's recombination. Swap strands readily. Crossover homologous regions. Mix them for me. This story is complete now. The DNA is fit for gametes now. The 3 R's for the DNA shine. Replicate, repair, recombine. Oh yeah. Replicate, repair, recombine. Okay, we'll stop there for the exam.
I asked the wrong question before, actually. I meant to ask, is it mutation that attracts
Is what?
Is it mutate that attracts the exon nuclease, or does mutate do the exon nuclease itself, not mutate? That's the question.
I'm not sure I understand the question, Lance.
In the mute SLH system, S binds, L is a bridge,